Want to start out just giving you a little bit of perspective. Our topic today is DNA, RNA, which collectively are called nucleic acids. And we'll also start talking about transcription and translation today and tomorrow. How many cells do you think are in an adult human? How many human cells are in an adult human? I just Googled this before class.
3 billion bajillion.
Yes, scientifically that's 3 billion kajillion, 37 trillion cells. That's a 37 followed by 12 zeros. The universe is about, 13 to 18 billion years old, so there's just massive numbers of cells in your body. This will gross you out. How many bacterial cells are you carrying around in your body? 38 trillion. There's, that part in is it 2 Kings, where Elisha asks the Lord to let his servant see the angels surrounding them. There are more of them that be with us are more than them that be with them. Well, it's actually backwards when it comes to bacteria. There's more of them in you than there is you in you. So it's kind of bizarre. Now, how much DNA do you have in your cells? The DNA is packaged in a couple places, your nucleus, and then mitochondria have some of their own DNA as well. There's about 30,000 genes in the human genome, about 3 billion base pairs in the DNA that's in your cell, one cell. If you stretch that out end to end, all of the double helix DNA, unwound it, it's tightly packed in something called chromatin. If you unwound it and lined all of it up in all of your chromosomes end to end from just one cell, it would be 6 feet high from end to end. That's just one cell, okay? It would be a tiny line so small that you couldn't see it, but end to end it would stretch out 6 feet. Now, if you've got 37 trillion cells, then 37 trillion times 6 is the distance in feet that your DNA could stretch. Trillions of feet. So I'm going to sneeze, sorry. Excuse me. So 6 times 37, whatever that is, more than 180, about 200 something trillion feet. Put that into perspective, that's about 120 billion miles. You know what the distance to Jupiter is? Almost 500 million miles. So you've got enough DNA in your body that stretched out end to end could travel from here to Jupiter and back 124 times. That's just kind of crazy. All right, so that's how much DNA you've got. The amount of length of the DNA isn't the important thing. The important thing is the information that's contained in DNA. And all of that's been summarized in what's called the central dogma of molecular biology. When I hear the word dogma, I think of a character from the Clone Wars animated series, Star Wars. But the central dogma or theme, doctrine of molecular biology is information in DNA it gets transcribed into an RNA message, which gets translated into a protein product, which does the work. So we're going to zoom in really close on DNA structure. We're going to talk about the nucleobases. We'll talk about the DNA double helix, and then we'll learn how to transcribe a sequence and how to read out what kind of protein that sequence will make. Before we do that, I'm going to stop the recording here because I want to show you, I'm going to screen record and show you something from a program called PyMol that allows you to see the three-dimensional structure of DNA. So, you guys, nope, you can't see that. You guys were looking at, oh well, there wasn't anything to show you so far. All right. This is a program called PyMol. It allows you to display the three-dimensional structures of proteins and nucleic acids. This is a DNA double helix. This is from a crystal structure. It's an experimental technique that we don't need to worry about, but you can actually see the positions of the atoms. All right? So just so you can get an idea of what's going on here, all the white spheres are hydrogen atoms. All of the gray spheres are carbons. All of the blue spheres are nitrogens. All of the red spheres are oxygen. And then the orange spheres are phosphorus. So this is a DNA double helix. You can see a couple of interesting things in this DNA double helix. You can see a spiral starting here. I almost feel like I need a laser pointer. Is that cursor good enough on the screen that you can see what I'm doing? Okay, this spiral represents this spiral that's sort of descending in this direction. Is you'll see it's got a phosphate group and then a bunch of carbons, another phosphate group, another bunch of carbons. The phosphate and the carbons that alternate, that's the sugar phosphate backbone of DNA. The sugar portion is ribose. We've drawn it before when we've drawn ATP, and you'll see it's the same sort of thing here. Attached to the sugar phosphate portion is a nucleobase, which I'm going to attempt to show you just by looking down the molecule. Let's see if we can, yep, that'll, okay. What I'm going to do is just, right, I'm going to hide everything except one pairing. Okay. And now I'm going to select this one, and we're going to zoom. All right, this is one rung of the DNA ladder. On the left, this portion is a ribose ring. This is actually the one of the OH groups on the ribose ring. And this is the 5', this is the 3' hydroxyl, this is the 5' hydroxyl here, and this is the phosphate group. Attached to the anomeric carbon of the ribose ring, you have this large structure. This is called a nucleobase. And you'll notice that that nucleobase is paired with another base on the other strand. And in between the two, you have hydrogens. These are called hydrogen bonds between the individual bases. And then on the other side, you have another ribose ring. So to understand DNA structure, you got to have in mind the sugar phosphate backbone. That's this portion out on the outer edges. And then you have to have the base pairs. You'll see in the overall structure of the molecule, I need to select it all. Show spheres. See in the overall structure of the molecule, the base pairs are in the middle, and the sugar phosphate backbone is on the edges. Another important thing to get from the structure of DNA is that you'll notice if you follow the sugar phosphate backbone down around the DNA duplex, there's two characteristic spacings between sugar phosphate backbones, okay? Where my cursor is now is one strand of DNA, and this is the second strand of DNA. There's a short distance between those two strands, and then if we rotate here, there's a larger distance between the two strands. The larger distance is called the major groove, and the smaller distance is called the minor groove. Yeah?
So on the other ones we were looking at with the beta strands and the beta turn,
Yeah.
does that one spin as well?
So yesterday when we were looking at peptides and proteins made of amino acids, we talked about beta turns and strands, and those twist as well, though the atoms involved are entirely different for a peptide versus a nucleic acid. So what we're going to do today is we're going to start with the basic structure of the bases. We'll learn about base pairing. I'll show you the structure of a base with a sugar phosphate attached to it. Then we'll expand to what it looks like when individual nucleotides, that is the base and the sugar and the phosphate, are linked together. And then we'll learn a little bit about how biology makes DNA and RNA, and we'll learn how to transcribe and translate. Okay, anything you want to ask about this three-dimensional structure? Yeah?
Do two strands ever cross over each other?
So the two strands, you can draw them like side by side like a ladder, and the rungs of the ladder are individual base pairs. The strands, like peptides, have directionality. We're going to talk about a 5' end of the strand versus the 3' end of the strand. That's not going to make any sense until I show you the structure of the sugar. But the two strands are actually oriented anti-parallel. They go this way with respect to each other. And then they twist around each other. Now, do they cross? I mean, yes, in the sense that they twist. But if you unraveled the DNA and untwisted it, it would be parallel. Okay, what else? Yes?
Wait, so what's the difference between the minor and major groove?
The major groove is this broad spacing between the sugar phosphate backbone of one strand and then the backbone of the second strand. But then on the other side of it, there's a smaller spacing. So interestingly, when the two strands twist, they don't twist in such a way that it's even spacing. There's a larger spacing in the twist and then a smaller spacing. Major groove and minor groove. And we're actually going to talk about that because when we're done today, you're going to see a piece of DNA like this. We're going to focus in on the major groove. You're going to see a base pair and look at the edge of that, and you'll be able to tell me what base pair that is. You'll say, oh, that's an AT pair. The minor groove is a little bit more difficult to read because it's deeper. It's narrow and deep, whereas the major groove is broad and shallow. These major and minor grooves are important because, and we won't learn about this at all, but there are things called transcription factors that bind in the major groove and recognize certain sequences and recruit polymerases that will then open up the DNA and start generating mRNA. This is how your body knows how to turn on a certain gene and express the gene product, is various proteins can bind in the major groove, recognize the base pairs, and say, hey, this is a gene that needs to be transcribed to RNA, and then that RNA needs to be expressed to become a protein. Good. Other questions? All right. Well, let's get started with the base pairs. All right. So let me draw for you a nucleotide, and we'll talk about the various parts of a nucleotide. So we'll start with the ribose sugar, and we'll do here a ribonucleotide, which means this is going to be based on ribose, and RNA is made of ribonucleotides. And we'll draw the nucleotide that you've seen before most commonly, and that's ATP. So as you've seen before in ATP, you've got alpha phosphate, a beta phosphate, and a gamma phosphate. And that's how we've sort of talked about these things. And then over here, we've often just abbreviated on the anomeric carbon of the sugar, there's a nitrogen. And that nitrogen's attached to what's called a nucleobase. And for ATP, the base is called adenine. It's got a 5-membered ring with this structure attached to the anomeric carbon of the sugar. And then over here, you've got a 6-membered ring with double bonds like this and an NH2 group. So this is ATP, and the complete name is adenosine 5' triphosphate, or ATP. What is this 3' and 5', and where does the name adenosine come from? First of all, this carbohydrate portion is called ribose, and then this portion of the molecule is called adenine. And it's, we represent it with the capital letter A. It's you don't want to get confused between A for adenine and A for alanine, so you sort of have to pay attention to context. The numbers of the carbons and nitrogens of the adenine ring are numbered. We won't learn that numbering system. It's not that hard, but it's just not useful in this class. We have to use a different set of numbers for the ribose ring. And so that's why we use 1' for the anomeric carbon, 2', 3', 4', and 5'. All right, so you will hear me talk about the 3' hydroxyl of a nucleotide. That's this one, as opposed to the 2' hydroxyl. It's the one on carbon 3 of the ribose ring. You'll hear me talk about the 5' oxygen of the ribose ring. That's the one that's attached to the CH2 group. Yeah?
Why is it prime?
It's prime because we're going to use numbers without primes for the atoms of the ring. I forget what the numbers are, but it's, you know, something like this. I'm wrong on the numbering scheme for the ring, but okay. So that's the adenine ring, and that's ATP. Questions about that structure? When you put adenine together with the ribose ring, that equals adenosine. And the naming system for the nucleotides is somewhat confusing, as you'll see in just a little bit. So adenosine on its own would be just the adenine and the ribose with just an OH group here. So I'll just copy that real quickly so that you can see. If it were just the OH group, we would call this adenosine. It's when you put phosphates on it that it becomes a nucleotide. Without the phosphates, it becomes a nucleus. It's called a nucleoside. That's a minor distinction that isn't going to be really important for us, but if you hear people talk about it, you'll know sort of what they're dealing with. Okay, other questions about the structure? Now, let's talk about deoxyribonucleotides. This is what DNA is made from. So I'm just going to copy all of this. And in DNA, it turns out that the 2' hydroxyl is gone. Your body deletes that 2' hydroxyl from ribose, and we call this deoxyadenosine 5' triphosphate, or this would be DATP. Your body's energy currency is ATP. Your body uses DATP to synthesize DNA through a mechanism that I'll show you. All right, so now let's talk about the 4 different bases. This portion, adenine, is an example of a nucleobase. And we'll now show you the varieties of nucleobases that are present in nature. So, there are two categories of nucleobases. It's fine if you think of them as bigger versus smaller. The first are called purines. That's a name you don't necessarily need to know, but the purines have 2 rings. And then the second class is called pyrimidines, and they just have the one 6-membered ring. So the first purine base that you've seen is adenine. It's got this 5-membered ring with 2 nitrogens on it. The nitrogen at the bottom is the one that's attached to ribose. And then the remaining 6-membered ring also has 2 nitrogens in it. And this is what it looks like, adenine. Now, let's look at the structure of adenine really briefly. We want to analyze the kinds of polar bonds and electron-rich atoms that are present in adenine. There are 2 kinds of groups in adenine. We have hydrogen bond, for HB is hydrogen bond donors. That is, there are atoms with, there are groups with NH bonds that are hydrogen bound to an electronegative atom such that there should be a delta positive, a partial positive charge on hydrogen and a partial negative on nitrogen. We call that a hydrogen bond donor, and as I label these nucleobases, I'm going to highlight hydrogen bond donors in pink. And then there are hydrogen bond acceptors, and these are essentially going to be electronegative oxygen or nitrogen with a lone pair. And in general, the lone pair is going to be non-resonance stabilized. So, looking at the structure of adenine, let's identify the hydrogen bond donating groups. Where do you see a hydrogen bond donating group on adenine? Yeah, you got these 2 NH bonds. Sometimes people call this nitrogen here the exocyclic amino group. It's exocyclic because it's outside the ring. It is not a hydrogen bond acceptor because here the lone pair is stabilized. That means energy lowered due to resonance. So the exocyclic amino group is a hydrogen bond donor, not an acceptor. Where do you see hydrogen bond acceptors for adenine? All the rest of those nitrogens that have lone pairs, if the nitrogen's already participating in a pi bond and it has a lone pair, that lone pair is in an sp2 orbital and localized, so it's not resonance stabilized. So yeah, these 3 nitrogens are all hydrogen bond acceptors. And you're going to see that the different nucleobases display a unique and different pattern of hydrogen bond donors and acceptors. Let's look at the other purine, which is G. G stands for guanine. Guanine is going to have the same basic core structure for its purine, for its nucleobase. That is, we got the same 5-membered ring with 2 nitrogens, and then a 6-membered ring with 2 nitrogens. But you're going to see that the nitrogens are a little bit different. In guanine, this nitrogen's actually an NH, and the carbon that used to have the exocyclic amino group in adenine is now just a carbonyl. And then the nitrogen below is doubly bonded, has a lone pair on it, and then has an exocyclic amino group here. So this is guanine, and let's highlight the donating groups, the hydrogen bond donating groups versus hydrogen bond accepting groups. Guanine has these hydrogen bond donating groups, and then it has these hydrogen bond accepting groups. Okay, any questions you want to ask about that? It turns out when you look at these bases, this side of the base is going to be the one that's involved in base pairing in a DNA double helix. So you can see that guanine has more donating and accepting groups. We'll call this actually, in honor of Watson and Crick, but not Franklin, unfortunately. It's weird how history gives credit to the men involved, but not the women. So maybe we'll call this the Franklin face of the DNA. In any case, this face of adenine and this face of guanine are, and I was alluding to a controversy about the discovery of the DNA double helical structure. If you want to know more, look it up on Wikipedia. We don't have time to tell you about it today. Guanine has more donating and acceptor groups that can be involved in base pairing. Okay, questions about guanine structure? And yes, I do want you to know these. There are 3 pyrimidines. One of them is for DNA, one of them is for RNA only, and one of them is for DNA only, and then the other is found in both. So let's deal with the weird ones. The one that's found in RNA only is called uracil. Its 1-letter code is U. And it's a 5-membered ring with 2 nitrogens. The one at the bottom is the one that's attached to ribose. There's a pi bond here, a carbonyl here, an NH here, and another carbonyl there. So in uracil, the hydrogen bond accepting groups are here and here, and then the hydrogen bond donating group is there. Then the one that's found in DNA only is the DNA version of U, whereas U is the RNA version of T. T is going to be exactly the same as uracil with one small addition. T is going to have a methyl group here. So that's T, and the name for the base pair for T is thymine. We'll have something to say maybe a little bit later, today or tomorrow. Thymine's important because it's one of the reasons why sunburn, why exposure to UV light causes DNA damage, and that's what actually is going on when you get a sunburn. And it specifically involves thymine. Okay, lastly is C. The name for C is cytosine. This is the one that's confusing, but we don't even really need to comment on the confusing nature of it. Normally, if a molecule has OS in it, we expect a sugar to be present. Cytosine refers to the base on its own. In any case, it's also a 6-membered ring involving nitrogens at these 2 positions. This one's the one that's bonded to ribose. Then you've got a pi bond here, another there, a carbonyl here, and an exocyclic amino group up at the top. So that's C, or cytosine. And again, if we label the groups that are donor versus acceptor groups, we've got hydrogen bond donors here and 2 hydrogen bond acceptors here. All right, those are the base structures. Now, if we take a step back and look at A and U, sort of from top to bottom down the what we would call the interface between base pairs, notice that A starts out with a donor and then an acceptor. Notice that U and T start out with an acceptor and then a donor. If you line them up side by side with their 2 Watson-Crick faces facing each other, they'd be complementary. They could hydrogen bond with each other. Similarly, if you look at the groups on guanine, on this sort of Watson-Crick face of guanine, you've got an acceptor and 2 donors, whereas for cytosine, you have a donor and 2 acceptors. Again, complementary. So what we'll do now is see if you can draw a base pair of A with, let's see what your quiz has you do. Is it U or T? I mean, it has you do T. So this would be in DNA. So all I want you to do is draw an A nucleotide, an A nucleobase. You can use an R for the ribose ring, and then a T nucleobase. And I want this NH2 group, hydrogen bound to this carbonyl oxygen, and I want this nitrogen on A, hydrogen bound to this NH on T. Okay? I want you to use dotted lines to indicate a hydrogen bond, sort of like we did last time with the beta sheet structure. And you may have to get started and erase to try to get it right. You'll have to probably, for T, in order to get this to happen, you'll need to flip it, draw it, pancake flip it, and draw it this way so that the 2 Watson-Crick faces are facing each other. I start by drawing one of the 2. You can do T on the left or adenine on the left. It doesn't matter which. Either's fine. And then I actually like to draw the dotted lines for the hydrogen bonds first. And then for T, I'll start with the hydrogen bond donating and accepting groups respectively. So for example, I know that an oxygen needs to go here, and I know that an NH bond needs to go here. And then I'm thinking, okay, how do I get the rest of T to fit into that space? And it's okay if you have to stop and erase and try again. I always do the one on the left better than I draw the one on the right. This is one of those you didn't know it was art class. Okay, questions about that? You should have a hydrogen bond between the exocyclic NH. Yeah, go ahead.
Maybe I just drew it wrong, but would you switch the methyl group and ribose groups?
Could you switch the methyl group and ribose? Yeah, this is a great point. You might have gotten basically the same thing. I'm guessing that what you're proposing is something like this, where this is ribose and then the methyl group is here. Is that right? It turns out that's totally reasonable, but that doesn't occur in DNA. You're seeing some flexibility with thymine. It's got 2 carbonyls on either edge and an NH in the middle, so it's somewhat symmetric. And without knowing what the structure of DNA is, you wouldn't know what it's supposed to look like. And the answer is in double helical DNA. Let's see if I can undo enough to get back to where I was. In double helical DNA, that's the way it is. The 2 ribose portions of the 2 rings are on the same side. That actually generates the major and minor grooves. This portion of the molecule, this side, this spacing between the 2 ribose rings is the wider spacing, and that's called the major groove, except I spelled it grove. Threw off my groove. Yes, you know what that's from? Yeah. That's the Emperor's New Groove is a Price family favorite. Amber and I saw that when we were dating right after my mission. We went and saw it in the Varsity Theater that used to show movies. Saw it several times, and then for years after that, it was our go-to movie whenever she was giving me a haircut. We'd turn on the Emperor's New Groove. We can quote basically anything in that movie, and our proudest moment as parents is when our children begin to quote the movie to us. For example, do I smell something burning? And one of the sons would say, my spinach puffs. And so that was great. You're quoting Kronk. Or a llama, he's supposed to be dead. And then the child would respond, yeah, weird. And again, proud parent moments. So maybe I can put some Emperor's New Groove quotes on the final exam. We'll see if we can come up with them. Okay, so down at the bottom, that smaller spacing between the 2 ribose rings is called the minor groove. All right. One important thing about an AT pair, or a TA pair for that matter, there are only 2 hydrogen bonds between those bases. And the other thing that you can notice about the AT pair is that despite the 2 groups that are hydrogen bound to each other, there are still a large number of unsatisfied hydrogen bonds. For example, on the major groove side of the base pair, you have this nitrogen, you have an NH that's not involved in a hydrogen bond, you have another lone pair on this carbonyl oxygen, and then also exposed to the major groove, you've got this methyl group, which can't hydrogen bond, but at least takes up space. So when we go back to the structure of DNA, one of the ways you can recognize an AT pair is to look for the methyl group and this pattern of acceptor, donor, acceptor. We might call this acceptor, donor, acceptor, methyl group, or atom. I have a son named Adam. I didn't turn out I just realized that right now. So if you see atom in the major groove, that's an AT base pair. It could also be MATA if you start on the methyl side, but yeah. Okay, questions about that? Go ahead.
So if the ribose bond was at the top where the methyl is, if it were there, there would be no groups.
Having the riboses face each other like this would be a different structure of DNA, and I don't know how to predict what it would look like.
Okay.
Yep, another hand. Yeah, somebody? Yes.
How do you distinguish if it's the major group or the minor group?
If you're looking at a base pair like this, the minor groove is the one where the riboses are closer to each other, and the major groove is the opposite side. Yep. That's not really predictable from this drawing, it just happens to be true based on the double helical structure of DNA. Sure.
So like if we're looking at an example and we're trying to determine if it's like true or false, is this correct? You know,
Right.
Would if it were the same distance between R on both sides, would that be?
Oh, so you're asking would I try to deceive you by drawing you something but the ribose rings aren't quite in the right position? No, double helical DNA with this kind of base pair sort of automatically results in a major and minor groove. You don't even have to do anything. Okay, let's do a GC one, shall we? This time, just for fun, let's put the smaller ring on the right, and then we have to draw the larger ring on the left. So I'll get you started on the C. So we'll draw the nitrogen, of the cytosine ring. We'll put the double bond here, the exocyclic amino group here, the ring nitrogen here, and then the carbonyl here. A hard time drawing slanted straight lines. All right, so to get going, you can draw your hydrogen bond lines, and they should all be parallel to each other. You're going to have 3 hydrogen bond lines, and then over here on the right, you want to start drawing your guanosine, or your guanine rather. The atom that hydrogen bonds to the NH of C should be the O of guanosine. The atom that bonds to the N of C should be the NH of guanine. And then the atom that bonds to the O on the bottom of C should be an exocyclic NH of guanine. So that's sort of how it's going to look. Now you got to connect the dots. Go ahead.
So why aren't we? So we started with A in the first one. Why aren't we starting with G in the next one?
We did that on purpose just to have, so technically there are 4 different kinds of base pairs. There's AT and TA, and then there's GC and CG. We're just doing half of them. So you could draw a TA base pair where you would put T on the left and A on the right. It would look basically like this, only you've exchanged the 2 positions. Yeah, we're going to draw the CG base pair, and then later on, if you want for practice, you can draw the GC base pair. All right, so the way to start filling in the dots is to notice with the structure of guanine, you've got O, then a carbon, and then a nitrogen. So the nitrogen sort of needs to go up and then up again to get the oxygen. So what I'd recommend to start filling out the ring of guanine is you go straight up from the nitrogen and then diagonal to the carbonyl oxygen. Then you can draw from there, you can sort of draw the 6-membered ring of guanine to the down to the left and then straight down to the exocyclic amino group, to the ring nitrogen. And then up here is where you're gonna put the 5-member ring portion of guanine. And I need a little bit of space. And this is the location of the ribose attachment. So this is C and this is G. Notice that the hydrogen bond donating group, exocyclic amino group of C, is hydrogen bound to a lone pair on the oxygen. Notice that the accepting ring nitrogen of C is hydrogen bound to this NH group. And then the accepting oxygen of C is hydrogen bound to the exocyclic amino group of G. So that's what a CG base pair looks like. And there should be a pi bond here. Both of these rings are fully conjugated. As before, this distance from R to R is the major groove. And this distance here, that space or that face of the molecule, is the minor groove. And you can see that the major and minor grooves display different things. For example, if we looked at the major groove for a CG pair, we would see a donor here with the exocyclic amino group. Struggling, there we go. A donor, we would see an acceptor here, the other lone pair on the oxygen, and we would see another acceptor there. So whereas the CAT pair is atom, the CG pair is da. Yes, go ahead.
So you can kind of mix and match them, but A and T have to go together and G and C have to go together because of how much hydrogen bonding there is.
Right. Yeah, A and T have to go together. They are complementary. A and T are complementary. They're always paired together. And then G and C are complementary. They are always paired together. This is really important. That's the basis of information storage in DNA, because that allows one strand of DNA to contain literally the instructions for making the other strand of DNA. If you know the sequence of one strand of DNA, you automatically know the sequence of the other one. And that's how cells replicate their DNA and then give them to daughter cells, because the instructions for the next generation of DNA are completely contained in a single strand. So yeah, there are AT base pairs, there are TA base pairs that we didn't draw today, there are GC base pairs that we didn't draw today, and there is a CG base pair. So let me just be explicit about that. This is an AT base pair. The TA pair is not shown, but you could draw it, and for practice you probably should. This is a CG base pair, and the GC pair is not shown. Okay, questions?
Is the major group always on top? Like if you were to draw GC, would the major group still be on top?
If you draw it this way with the major groove is always on the opposite side from where the ribose rings are, however you choose to draw it. Okay, so what I want to do now is just practice a little bit, not drawing the base pairs, but actually looking at some double-stranded DNA and seeing whether you can read the sequence of the base pairs from, oh, dang it, just wanted copy, there we go. See if you can read the sequence of the base pairs from what's in the minor groove. I'm going to erase the answers to delete, 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 delete. Okay, shown here is a schematic version of, and this will be in the notes that I post, so don't worry about scrambling to get it down. A schematic version of DNA and then also a crystal structure or a model of DNA structure. This shows you a cartoon where the bases are different colors and the sugar phosphate backbone is this blue ribbon. And you can see in DNA structure that the sugar phosphate backbone starts at the 5' end, goes to the 3' end. I'll have more to say about that in a minute. That's one, and then the other one starts at the 5' end and goes to the 3' end. The arrows indicate 5' to 3' direction. The two strands are opposite or anti-parallel each other. And you can see that the bases are complementary to each other. Here is an AT pair. This is a TA pair. Here is a CG pair. Now this is a GC pair. And that complementary pairing is important. Notice that as we've drawn them, the AT and GC base pairs, or vice versa, all take up about the same amount of space. There's always a purine paired with a pyrimidine. You never pair a pyrimidine with a pyrimidine because the space between the two of them would be smaller and they wouldn't fit neatly into the DNA structure. Nature uses both the hydrogen bonds between AT and GC pairs and the overall shape to determine which base needs to fit opposite the corresponding base in the other strand. All right, here's double helical DNA, and we're looking in the minor or the major groove. So I would like for you to remember carbons are gray, nitrogens are blue, oxygens are red. So our pink and blue color scheme might actually be unfortunate, I don't know. But, and then carbons are gray, hydrogens are white. I want you to look at this, focus in on this particular base pair. I want you to look at the groups displayed in the major groove, and I want you to tell me what kind of base pair is this. You've got four choices, GC, CG, AT, or TA. Colors are atoms, so gray equals carbon, red equals oxygen, blue equals nitrogen, and white equals hydrogen. Orange or yellow equals phosphorus. You can ignore the phosphorus for our purposes here. So the base pair in the box, what do you think it is? What do you see?
I'm not doing well on the quiz.
You're not doing the one on the quiz yet. We're practicing. Go ahead.
Is it CG?
Is it CG? Which one?
Left to right CG.
Left to right CG. Let's see. Ha. That's not that funny. You got it? Okay. It was mildly amusing. It was worth a chuckle. Yes. How can you tell this is C?
Well, I just looked at it and saw if I could see a methyl pointed out, and I didn't. So
Okay.
I figured it wasn't TA.
Okay.
And then I just looked on which side the carbonyl oxygen was, and it's on the right side.
Okay. So let's go back to our base pairs and review what's in the minor groove for each of them, major groove for each of them. The key dead giveaway for a TA base pair is the methyl group. Okay, so that's easy. But then it should have, in addition to the methyl group, it should have an acceptor, donor, acceptor. For a CG or GC base pair, it should have two acceptors and a donor, one NH bond, an oxygen, and a nitrogen. And the NH bond is always on the C side. That was sort of punny too. We could think of a little song about being by the seashore. Yeah. So This group here is the NH from a C base pair. This is the hydrogen, this is the nitrogen. That's the NH bond. And that's an acceptor, right? Or that's a donor of hydrogen bonds. Then here is the oxygen that's doubly bonded to a carbon, and then this is the ring nitrogen from G. So this is a CG pair, and these two are hydrogen bond acceptors. You see how we did that? Just looking at the edge. In order to get this kind of question right, you have to basically have in your head or drawn on paper in front of you the structures of an AT versus CG base pair. If you've got that, then you can figure it out. Go ahead.
Would you put a question like that on the
I would have and will. Yes, would I put a question like that on the exam? I have. In fact, this question from your quiz is lifted from a previous exam. So let's look at that now in your quiz, and let's give you a chance to work on that. So this is another structure of DNA, and you're just going to zoom in on the one in the box, look at the functional groups that are exposed in the major groove, and again, your goal is to tell me which of the four possible base pairs left to right is it? GC, CG, AT, or TA? This is called learning to read like a transcription factor. These are the proteins that bind to the major groove and recognize these things. Yeah?
Hypothetically, if someone was colorblind, how would they do this problem?
Hypothetically, if someone was colorblind, how would they do this problem? Let's think about that. It depends on the kind of color blindness. If it's red-green, there's no other green in the structure. It would be hard to solve in black and white. If that's your situation, why don't you come talk to me and we can talk through what would make this problem doable. I'm not sure. In my research publications, I try to avoid red and green and yellow-blue combinations exactly for that purpose, but I'm not exactly sure what would work here other than labeling all the atoms, which are supposed to be done. In this case, shape could help you because even if you're red-green colorblind, you can probably recognize what's the dead giveaway here. Methyl group, right? You see the methyl group, it's got to be an AT or a TA pair, and the methyl group's always on the side of the T. So that's easy. Then you can confirm that it's a TA pair because in a TA pair, as we talked about, or an AT pair, it should, M is for methyl, it should be atom, acceptor, donor, acceptor, methyl. And it is. This is a ring nitrogen. This is an NH bond. That's a donor. So acceptor, donor, carbonyl oxygen, acceptor, and then methyl. So this side, the methyl side, we'll call it ADAM. The methyl side is always T, and then the other side is A. Okay. And actually, with what's on the quiz and what'll be in the notes, you can probably look, the view isn't perfect, but you can look at some of the other edges in the major groove to see if you can tell what those are, and that might be useful practice. Go ahead.
Do we need to know how to do this with the small group, the minor group?
Do you need to know how to do this with the minor groove? No, because it's too deep to see in a picture. Though some chemists have invented molecules that bind in the minor groove and do sense the functional groups that are there, and it works pretty well. Okay, we need a break, so let's take 5 minutes or so, and when we get back, we'll just continue with DNA structure.